Hi students, this is going to be the fourth and last part of the cardiovascular system, and here during this portion we're going to be talking about the different abnormalities of the cardiovascular system. So we'll start off with hypotension, and hypotension literally translates into what it means is a low blood pressure, okay, regardless of what the cause of hypotension is. So hypotension could be due to significant loss of blood. So somebody that had a lot of bleeding, these patients would have what is known as hypovolemic hypotension. So they lost a lot of their blood volume. Other causes could be due to allergic reactions, for example, where there is massive vasodilation of the blood vessels, and that leads to hypotension as well. It could be due to heart failure, like cardiac decrease in cardiac contractility, or very strong emotions. So we'll talk about, the three types, the scientific terms for all of these different types. And hypotension, if it is severe, it can lead to shock. And shock is really a drastic decrease in blood flow to the different tissue. So we have three different kinds. There's hypovolemic shock, and that means significant decrease in blood volume. And that could be due to, severe bleeding or severe dehydration. So there's loss of fluid, and that led to severe dehydration and severe hypotension, leading to this hypovolemic shock. It could be low resistance shock, and that's what happens in severe allergies, where again, there's massive vasodilation leading to a sudden severe decrease in blood pressure, leading to strong enough to lead to shock. Cardiogenic shock is a shock that happened due to a defect or a pathology in the cardiac muscle. So it could be due to heart failure, somebody that had a heart attack, or myocardial infarction. And so shock is really a severe hypotension that led to a massive decrease in the blood flow to the important tissues, and sometimes may even lead to tissue damage. This is the distribution of the cardiac output to the different organs at different times. So we on average, we have about 5 liters of blood. And this is the distribution of those 5 liters at rest. So you'll see that a good portion of it, about a quarter of it, goes to the abdominal organs, and about 20% would be for the kidneys. But under exercise, you'll see that this distribution is going to change. So you'll find out here that more of that blood is going to be shifted to the skeletal muscles, less, that 24% goes down to 3% to the abdominal organs, and there is just a total shift of that, of the blood to different organs, and that's by either the vasoconstriction of some blood vessels versus the vasodilation of others. So how is it that the cardiovascular system has that kind of control in order to make these changes during exercise? So it's basically up to 2 different things. We have the local changes, so the local buildup of metabolites, and the local hypoxia, relative hypoxia, since now the muscles are contracting more and they need more oxygen. So these local changes are going to lead to vasodilation of the arterioles in the parts that are needed. So again, your skeletal muscles, for example. While the autonomic nervous system is going to make changes to the heart. So decrease of the parasympathetic output plus the increase of the sympathetic output to the hearts and to your blood vessels is going to lead to tachycardia, will also lead to increased force of ventricular contraction, and that will lead to an eventually increase in cardiac output. It is also going to constrict the arterioles that are at this point not very important when we're exercising. So we'll constrict the blood vessels going to the digestive system, as we've seen in that previous slide, and we'll kind of shift that blood from abdominal organs or less important organs at this point during exercise to the more important ones, which would be your muscles. So again, the control during exercise is up to 2 different things: your local metabolic changes plus the autonomic nervous system. This chart, or these charts, compare cardiac output stroke volume and heart rate between a trained individual, so an athlete in green, and an untrained individual in orange. Okay, so you'll see here that the heart of an athlete is healthier and therefore can produce more work or can produce higher cardiac outputs with less work. So basically that it is so, it has the ability to perform the same amount of work and even better, okay, but using less effort. Okay, so we see here that this untrained heart is only able to increase the cardiac output, to a certain degree, but the trained heart can increase the cardiac output even more, and that is because the heart of an athlete is stronger, has adapted to strenuous exercise, and is able to do that without putting too much strain on the heart. So you'll find here that if you have two different people, one trained and one untrained, the athlete, as they exercise, the heart rate of the athlete, whether at work or at rest, is always going to be less than the untrained heart, meaning that this trained heart can do the same work, okay, at a lower heart rate, which means that they are healthier than this untrained one. As for, stroke volume, you can see here that the trained heart, again, even because it has that lower heart rate, the ventricles are able to fill up with more blood, and when they contract, they produce a bigger stroke volume. Compare that to the stroke volumes coming out of this untrained heart. So again, the hearts of trained athletes are capable of maintaining same cardiac outputs, maybe even better, with slower heart rates and larger stroke volumes. Now, hypertension, that is the increase in blood pressure, the systemic blood pressure. And hypertension is one of the risk factors of heart attacks and renal damage, and has a whole list of complications. Now, the two major causes of hypertension, it's either primary cause, and that is the most common cause of hypertension. We used to call that essential hypertension, which is basically due to, advanced age. Risk factors are like diabetes and smoking, bad dietary habits, sedentary lives, and so on. While secondary hypertension is secondary to another disease. So it could be that this person has maybe kidney failure, and that led to the secondary hypertension. So secondary hypertension is secondary to another disease. Okay, so in order to treat this one, you would have to treat the initial diagnosis to control that hypertension. So contributing causes of primary hypertension. Okay, so again, these are your primary, which is the most common cause. We've got all of these are risk factors: obesity, diabetic patients, the insulin resistance one, so the type 2 diabetes, not the juvenile, but the adult onset, chronic high salt intake, stress, smoking, excess alcohol or caffeine consumption, poor diet, low birth weight, and not being breastfed as an infant. Those long-term were found to be risk factors for developing primary hypertension later on in life. And then genetic problems with the renin-angiotensin-aldosterone system. Okay, primary hypertension can be managed by, healthy diets, healthy exercise regimens, and lifestyle changes. Sometimes we have to introduce some medications. And we'll talk about the secondary hypertension. Okay, like I said, this is secondary to another disease. So in order to treat this, you want to treat the actual cause. So kidney damage is one of the main causes of secondary hypertension, and we call it renal hypertension. Or it could be due to the hypersecretion of cortisol, aldosterone, or thyroxine. So if you go back to those hormones, we talked about how their increase can contribute to high blood pressure. And And then even abnormal sleeping patterns, okay, and sleep apnea, those are risk factors for developing secondary hypertension. So we'll take a look at the kind of medications that we could give for either kinds of hypertension. Okay, these are the common ones. So diuretics, okay, diuretics that is going to increase the excretion of sodium. So when you dump more sodium into the urine, by osmosis, it is also going to pull water with it. Okay, and that is going to reduce the overall blood volume and pressure. A beta adrenergic receptor blocker or a beta blocker. So remember that the receptors on the cardiac muscles were beta adrenergic receptors. If you block those receptors, you can decrease the cardiac output, which will eventually lead to the decrease in blood pressure. Or we could use calcium channel blockers, and those are going to block the calcium channels found on the smooth muscle fibers on your blood vessels. Okay, and that leads to vasodilation. Okay, so we've decreased the peripheral resistance, and that decreases blood pressure. Or we can use ACE inhibitors, which is angiotensin converting enzyme inhibitors, and that blocks that whole angiotensin II production by that renin-angiotensinogen story that we've talked about previously. So if you block that, you're also blocking aldosterone, and that leads to the increase of excretion of sodium and water. And all of that eventually will lead to the decrease in blood pressure. You don't use all four of these for patients. Patients tend to respond to one versus the other. So there is a whole regimen on which one to start with and so on, depending on the cause, which we're not going to get into. But I just want to make it clear that you don't give all four of these medications to your patients. Now, coronary artery disease, that is obviously, hypertension is a risk factor for coronary artery disease, which would be, atherosclerosis that has happened in the coronary arteries, and that can lead to ischemia or decreased blood flow to the heart. This eventually can lead to a myocardial infarction, which is gangrene of the heart, or we also known as a heart attack. So heart attacks, the number of Americans that have a heart attack each year is about 1.1 million. About 40% of these end up dying because of the MI. Some of the reasons of death would be due to massive myocardial infarction. So the coronary artery disease, or that, whether it was a clot so that blood clot kind of led to massive gangrene or massive death of a good portion of the cardiac muscles, or it could have led to VFib or ventricular fibrillation, where the ventricles are contracting at a very, very high rate, and pretty much it looks like the heart is shaking as opposed to actually filling up during diastole and then contracting during systole. Because of the speed of it, there is really no cardiac output coming out of the heart anymore. Small fraction of patients that do enter into VFib, they can be resuscitated by using CPR measures and defibrillation. So, cardiopulmonary resuscitation and defibrillation. When you do this, though, you want to make sure that they also get a blood thinner, because they could have developed blood clots during the fibrillation time, and that could, those blood clots can be dislodged from the ventricles when it starts to contract and be lodged into different parts of the body. Now for hemostasis, hemostasis means to stop bleeding, okay? And this is a function of your platelets, and it's made out of 3 different steps. There's vascular spasm, and then the second step is formation of a platelet plug, and the third step is blood coagulation, and they happen in this order. So, you cannot really start off at blood coagulation. You have to start at vascular spasm, and then step number 2, and then 3. So we will talk about them in that kind of order. So vascular spasm is where the injury happened, and there is now bleeding. There are different theories on why the blood vessels actually constrict. Some of them might be triggered by the pain. It could be triggered by the chemicals that come out of the injured cells and those dead cells now. So we really don't understand completely what happens, but or why it happens. So these blood vessels are going to constrict, and that decreases the amount of blood loss. Okay, the vascular spasm, though, does not last for a long time. So at the most, it'll last for about 30 minutes, and then the blood vessels will dilate back to normal. And that's why we have to, those 30 minutes is enough time to move on to step 2, which is the formation of your platelet plug. Okay, and this vascular spasm is more effective if the injury is small. So when you have smaller blood vessels that have been injured, vascular spasm would help in decrease of blood loss. But if you have bigger blood vessels, say, the aorta, for example, that was injured, vascular spasm will have a very limited effect to stop bleeding. Now, formation of a platelet plug, under normal circumstances with a smooth endothelium without any damage, the platelets do not aggregate. They don't, they're not activated, and they do not stick together in order to make a platelet plug. When we lose that endothelial lining, and now these platelets are exposed to the connective tissue, whether connective tissue in the blood vessel or connective tissue in the, outside of the blood vessel, that is going to lead to the activation of these platelets. Okay, so when platelets are activated, that occurs by these different chemicals like ADP, serotonin, or thromboxane A2, and all of these are chemicals that are released due to the injury. Okay, those are going to activate your platelets, and by positive feedback, these activated platelets are going to now activate more platelets, and all of them are glued together in order to make this platelet plug. And that gluing of the platelets together is known as platelet aggregation. So you can see here, the prostacyclin, which is the PGI2, nitric oxide, thromboxane A2, all of these are coming out due to the injury and leading to the activation and aggregation of these platelets. And again, it'll lead to a platelet plug that will temporarily plug the site of injury. And that, again, is a very temporary thing where very fragile, but it's there to kind of temporarily stop the bleeding until an actual blood clot is formed. Now we'll get to the formation of the clot. Okay, so blood coagulation or blood clot is making that thrombus, which is a solid-like gel, and it basically looks like, I'm just going to go to the picture to show you what it looks like. So we have these little threads of fibrin, and those fibrin threads, or that mesh, acts like a net, and then that net is going to trap bigger cells like red blood cells and so on until it makes a blood clot. So your blood clot is just fibrin threads with blood cells, especially RBCs, that have been trapped by those fibrin threads. Know that fibrin, we normally have fibrin in an inactive form known as fibrinogen. And fibrinogen is made by the liver. Okay, so if somebody has liver failure, they would have a deficiency in fibrinogen, and that could lead to the loss of the ability to clot, which basically means that they have, they're very prone to bleeding. Okay, and fibrinogen needs to be activated into fibrin, and that could be done in 2 different pathways. Okay, so take a look at these 2 different pathways. It's either an intrinsic pathway or an extrinsic pathway. And these pathways don't mean that this is happening inside of the blood vessel and this is happening outside of the blood vessel. Okay, so they're both happening at the site of injury. The intrinsic and extrinsic just refers to the very first step where there's a whole cascade of events that need to happen. So we can't really just activate fibrinogen into fibrin. Our body makes it very hard to do that, because if it was an easy thing to happen, we would be developing blood clots all the time. So there's a whole cascade, kind of like a domino effect, where all of these steps have to occur, where at the very end, we're going to be activating fibrinogen into fibrin. And all of these different steps, we're activating different clotting factors, okay? And all of them, or most of them, are made by the liver as well. So you can see why liver damage would lead to bleeding tendencies. And again, the name intrinsic and extrinsic comes from the fact where that very, very first clotting factor, was it found inside of the blood vessel, or was it found inside of the tissue factor, which would be outside of the blood vessel? So a tissue factor, activation of the first step, which is a tissue factor in this side, and that's why we call it an extrinsic pathway, while an activation of the first protein found inside of the blood vessels, that would lead to what is known as the intrinsic pathway. And I don't want you guys to worry kind of about this whole cascade and, you know, which one activates which and so on, but you really want to focus on this, what is known as the common pathway. So both of these are going to lead to this bottom part known as the common pathway. And I thought it was here somewhere. Okay, where I'll go back to those those slides in a minute, where we are going to activate. No, this is not it either. So I went ahead and kind of got this slide from a previous course of yours where it shows the common pathway. And so in order, these are the last steps where prothrombin, which is an inactive form, would have to be activated into thrombin. And that is through an enzyme known as the prothrombin activator. Now, this active form of thrombin is what activates fibrinogen into fibrin. And then fibrin is going to eventually make the blood clot. Okay, so that is how that common step works, or that common pathway works. Now we've talked about, you know, all of these different things that are made by the liver, so we kind of want to focus on the role of the liver in blood clotting. It is going to make all of the clotting factors, okay? So all of your clotting factors, whether intrinsic or extrinsic or common pathway, they're all synthesized by the liver. It also makes bile salts, and bile salts are important for the absorption of vitamin K. And vitamin K are needed to synthesize these clotting factors. So not only does the liver make your clotting factors, but it also helps via the production of bile in the absorption of vitamin K, which we need to make these clotting factors. Now, as we mentioned, clotting factors are produced by the liver, and they are all secreted in an inactive form. So we do not want these clotting factors to be circulating in our bloodstream where they are already activated, because then we will be making blood clots. But they're all made in inactive forms, and you've seen how the cascade works, where each one activates another one. It's kind of like a domino effect. And at the end, if you take plasma and you use all of these clotting factors, that plasma is now going to be called serum. So serum is basically plasma minus the clotting factors. Now, what are the factors that oppose clot formation? There are a couple of different things, meaning, why is it that we don't really make blood clots? We have what is known as tissue factor pathway inhibitors. These are that's a chemical that is made by the endothelial cells, and it prevents the activation of platelets, and will prevent the activation of your clotting factors. Thrombin has a receptor on the endothelial cells known as thrombomodulin. Okay, and that would wants to tone down the effect of thrombin. Okay, and that thrombin is going to then attach to protein C and activate that. When you activate protein C, that is going to inactivate other factors. So basically, thrombin is one of the clotting factors that you have to activate in order to start making a blood clot. But to prevent too much blood clotting, it has a way to downregulate the formation of blood clots, and that is through the binding to a protein known as protein C, and that is going to deactivate the, some of the blood clots or some of the clotting factors. Antithrombin 3, okay, that is another way to deactivate or inactivate thrombin, and again, kind of tone down how much blood clotting occurs. So yes, you do want a blood clot to happen to stop the bleeding, but there is a point where you no longer need that. So these are two factors that help in, stopping the process. So how is it that thrombin, indirectly deactivates or inactivates some of the clotting factors through this protein C? So the endothelial cells has that receptor known as thrombomodulin, and you can see from the name of the receptor what it does. It modulates, it modulates the function of thrombin. So thrombin is now going to be to attach to protein C, activating it. So now this activated protein C will deactivate these two other clotting factors. That we have made the blood clot, there has to be a point where we get rid of that blood clot, and that is through the process of what we basically want to do is make that fibrin that was insoluble and made a mesh. We have to make it soluble again in order to disintegrate it. So this is the process that we do. We have a built into the clot, there is plasminogen. The plasminogen is going to be activated by these tissue plasminogen activators, or TPAs, into plasmin, and plasmin is going to disintegrate fibrin and make it soluble. Okay, and now we're able to break down that blood clot. So you'll see this plasminogen activators, one of the most common is tissue plasminogen activator that converts plasminogen into plasmin. And again, plasminogen is actually embedded within the blood clot and it is there just waiting to be activated. So when a blood clot gets way too big and there's just way too much fibrin, that is going to activate these TPAs, okay, and activate that whole process. And now we're able to make that fibrin into soluble parts, and we can break it down in order to get rid of that blood clot. That, happens pretty much, later. We can speed it up a little bit, especially if somebody has like a stroke. In order to dissolve that blood clot, we can give them synthetic TPA to help with that process. Now, what are the role of coagulation factors in clot formation disorders? So we kind of talked already about the effect of having liver failure and how the liver is responsible for producing all of the clotting factors and helps in the absorption of vitamin K. So that, we definitely liver failure patients will have bleeding tendencies. Now, these are other things. So hemophilic patients, this is a genetic disorder where there is, a pathology in the genes that are for the specific coagulation factors. So again, it's a bleeding disorder. Von Willebrand disease. Von Willebrand is one of the clotting factors. So in the von Willebrand disease, there is a reduced levels of that von Willebrand factor. F here stands for factor, and that leads to the decrease in the ability to make a platelet plug and then eventually make a blood clot. Patients that have vitamin K deficiencies, okay, remember that vitamin K is needed by the liver to synthesize clotting factors. So if anybody has deficiency in vitamin K, that can lead to the inability to make these clotting factors, and hence again, these patients would have bleeding tendencies. And now that concludes chapter 12.